All right, so glycogen is the animal energy storage molecule. This is the fuel storage. This is all alpha glucose. Structurally, it is very similar to amylopectin, but it is more densely packed. There is more branches of the alpha-1,6 linkage that we discussed before. That alpha-1,6 linkage, I am going to simplify that drawing. All right? That will be our 1,6 linkage. That is our alpha-1,6 linkage. All right? That's our alpha-1,6 linkage. And the whole thing will continue from there. So we have more branches, more closely packed, and we have shorter chains by right of them being more closely packed. So if you have more branches at greater frequency, you will have shorter chains of alpha-1,4 glycosidic bonds. So let's talk about the structure of cellulose now. We discussed earlier with our beta glycosidic bonds, with our beta-1,4 glycosidic bonds, where we have the up, the down, etc. So because of this, we will have a hydrogen bond here. Oops. CH2OH, CH2OH, versus A1 CH2OH. So here we have this hydrogen bond there, and we also have this will be our all right. See, we have the OH, 2 hydrogen bond there. Hydrogen bonds, remember, are always described as are always written and drawn as a dashed line. All right. So this alternating pattern of up, down, up, down, up, down, up, down, up, down, allows for these chains of beta glucose to hydrogen bond to one another. The tightness of these hydrogen bonds effectively, well, it's a branded product, but whatever, Ziploc sealing to one another, is going to prevent water from being able to leak through a fiber. A cellulose microfibril will be water insoluble. Beta glucose cannot dissolve in water. Water cannot pass through these tight junctions between these hydrogen bonded fibers of cellulose. But the individual microfibrils are going to be arrayed in such a way that they will effectively be full of holes. So cell walls, which are primarily composed of layered microfibrils of cellulose, are going to be full of holes. They're going to be porous. So they will be freely permeable to water, and water can add here, water hydrogen bonding to a substance that is not water, to cellulose microfibrils, in order, and this allows for the apoplastic pathway when we discussed Unit 4. It also allows for the transpiration, adhesion, and cohesion forces later on, also in Unit 4. All of this stuff will be seen again. Now, typically, a carbohydrate monosaccharide are those with ratios of C1H2O1. This is what I told you. But you can dope in the sense of metallurgy, where you add a carbon and iron to make steel, for example, carbohydrates with other elements to change its properties. For example, adding nitrogen to a carbohydrate will give you N-acetylglucosamine. N-acetylglucosamine, which has a chemical formula of C8H13O5N, that repeats N times. You can have this unit repeat multiple times. This polymer gives you chitin. Chitin is the shell material in crabs, in beetle arthropods, in any insect or arachnid or whatever that you find. This is going to be N-acetylglucosamine. So that's N-acetylglucosamine. That is a carbohydrate. Glucosamine is a carbohydrate, but it has nitrogen in it. That can be a question. Do not be fooled. Now let's talk about lipids. Amphipathic lipids are those lipids that have a polar head and a non-polar tail. Fatty acids will have the formula CnH2nO2. These are carboxylic acids with a hydrocarbon tail of variable length. Attaching a compound to the carboxylic acid end will give you soaps and detergents. So if I add length to this tail, so if I make this a hydroxyl group here, and I make this a carbon tail length that just continues on and on and on like that, that's fine. If I shorten the tail like that, that's also fine. Whatever, it's still a fatty acid. There are two types of fatty acids in this world. There are saturated and unsaturated. All it takes to be unsaturated is to have a single double bond in your hydrocarbon chain. By single double bond, I mean you only need to have one, how to phrase this without using the word single. You only need to have a double bond found somewhere within the hydrocarbon chain. That's a better phrasing. Let's use that. Saturated. A saturated fatty acid is one where there are no double bonds. Every carbon is bonded to at least two hydrogens. At least two hydrogens are bonded to every single carbon. If you have a double bond, these two carbons here that are participating in that double bond are only bonded to one hydrogen. It is not possible for them to bond to anything else. So that is a saturated and an unsaturated. Unsaturated. They could be saturated if you had more hydrogen. A saturated towel is one that cannot hold any more water or fluid or whatever. So a saturated hydrocarbon chain is one that cannot hold any more hydrogen, no matter what. Now, your saturated fatty acids are going to be your butters, your eggs, your meats, your fishes. Your unsaturated fatty acids are going to be your oils, your fruits, your vegetable oils, all those things, your peanut butters, which is also a vegetable oil, by the way. So, which is greater? The melting point of a saturated fatty acid is higher than an unsaturated fatty acid. This has to do with a property in organic chemistry of similar to like dissolves like, where polar compounds will fit better together if they are unsaturated. But as soon as you put a double bond in that chain somewhere, eventually what you're going to end up having is you're going to have more space right here, and they're going to separate more, and they're going to have less of a van der Waals force interaction, and less interaction between molecules means that they will separate more easily at lower temperatures, which means they will have a lower melting point. So an unsaturated fatty acid will have a lower melting point. The density of a saturated fatty acid you can fit more of these fatty acids together if they are saturated than you can unsaturated fatty acids. These will take up more space because they are bent and kinked. Reactivity. Saturated fatty acids are less reactive than unsaturated fatty acids because double bonds between carbons are more reactive from a chemical standpoint. But therefore, when you burn them, when you use a chemical reaction combustion to burn saturated fatty acids like fat, whale blubber or whatever, kerosene, for example, it burns very well. A very high energy, a very high temperature flame. So you will have a higher reactivity, got that backwards. It'll be a higher temperature flame with oil than it will be with whale blubber. You'll have a lower intensity, longer burning flame with whale blubber than you will with a oil. So a grease fire will be more energetic than will be a penguin blubber fire. And by the way, yes, you can actually have a fire with nothing but penguin meat. It is that fatty and oily. It is terrible. It's supposed to be very bad tasting. I've never had it myself. But conversely, when you metabolize it, when you yourself eat these substances, you get more energy. You get more ATP by using saturated fatty acids as fuel for aerobic metabolism, aerobic respiration, than you do for unsaturated fatty acids. And the reason for this is that your body, your living things, your living cells, they are going to utilize energy from carbon-carbon single bonds. The proteins and enzymes that are in your cells are set up for the breakdown of carbon-carbon single bonds. So you get more energy more efficiently from saturated fatty acids than from unsaturated fatty acids. So calorie for calorie, unsaturated fatty acids are better for you. That's why they keep saying that plant fats are better for you than animal fats. Why you should eat more plants than animals. Now let's talk about nonpolar lipids. Those were amphipathic lipids that we just talked about. Nonpolar lipids are things like waxes. These are esters of fatty acids. If you remember, an ester is a carbon-oxygen-carbon bond. So, carbon, oxygen to carbon. So you have your carbon chain over here, your carbon chain over here. A wax will have that going on over there, but steroids can be recognized by their tetracyclic skeleton, which means 4-ring skeleton. They are consisting of 3 fused 6-membered rings and 1 5-membered ring. So one, oh wow, that looks terrible. Let's fix that. Nope, that is also terrible. Make that look good. All right. All right. There we go. Other stuff will be attached to it, but that other stuff varies and is not something you can be tested on directly. So, this is our wax. Blah, blah, blah. Now, we also have triglycerides. Triglycerides are a non-polar fatty acid. Effectively, you take this, and you're going to get this as your simplified diagram. This simplified diagram, you have your glycerol. Glycerol is carbon, hydrogen, hydrogen, carbon, hydrogen, carbon, hydrogen, hydrogen, hydroxyl, hydroxyl, hydroxyl. Bonded to three of those. Does this look familiar to you? I certainly hope it does. Condensation reaction will form this. It will be formed by a condensation reaction. Water will leave and you will be left with a carbon-oxygen-carbon double bond, an ester, forming a fatty acid. A glycerol with 3 fatty acid tails. This will happen 3 times, you'll get 3 waters. Now, triglycerides. These are the storage forms of lipids in animals and plants in the forms of fats and oils. The food items that were shown as examples of saturated and unsaturated fatty acids were actually examples of fatty acids clumped into triglyceride units. So, in animals, your triglycerides will typically be saturated triglycerides. And your plant oils will be unsaturated triglycerides. They will consist of unsaturated fatty acid tails on a triglyceride, glycerol with 3 fatty acid tails. They are a fantastic source of energy. They are a fantastic source of fuel because they have many carbon-carbon single bonds. They are highly reduced, so you can release this energy by oxidation. They can be localized in an organism due to their insolubility in water. Because they are not polar, they will not dissolve in water. They will not easily diffuse throughout the cell. They will stay wherever you put them. So, if you have a cell and you want to store triglycerides, if you have your cell over here with your nucleus over here, and you want to store triglycerides, you can make a clump over here, unbound by a membrane, and just have a clump of triglycerides. Not Cambridge appropriate, because I labeled inside the diagram and I shaded. Unacceptable. So, this doesn't even need to be bound by a membrane inside the cell. They will clump together because nonpolar will dissolve nonpolar. It will clump together. Like dissolves like. And the polar water molecules around it will form this bubble. So you can just sequester triglycerides within a cell. You don't need to do anything special to store them. So, it's a condensation reaction. Well, I've already said this. It's 3 condensation reactions, actually. Now, how do we turn a triglyceride into an amphipathic lipid again? We add a polar group. So, we have 2 fatty acid tails, and we have a phosphocholine group. This is the beginnings of our phospholipid. Our phospholipid is extraordinarily important when we'll start discussing in Unit 2 the plasma membrane, which is primarily composed of phospholipids. So here, we'll have our glycerol as our backbone. We will have 2 fatty acid tails, whether they are saturated, as shown here, or unsaturated with kinks in the chain here and there. Doesn't matter right now. We're not working with that. And one of those spaces will have a phosphate group, which is negatively charged, and a choline group, which is positively charged. having a positive and negative charge in one clump. This is a polar molecule which has a positive and negative end. That makes this a polar molecule over here and a non-polar set of molecules over here, but they're attached as one molecule. So you have a polar head and a non-polar fatty acid tail. Polar phospholipid, polar phosphorylcholine head, non-polar fatty acid tails. Cholesterol. We can add to a steroid compound, that tetracyclic ring, a simple hydroxyl group right here. And suddenly this whole molecule is polar. This one group, which has an electronegative charge, which has electronegative difference in charge, from a ability to form hydrogen bonds, makes this a polar head and this a non-polar tail. So what does that really get us? It allows us the ability for water to hydrogen bond with the polar groups of cholesterol and the phospholipid heads on either side of a phospholipid bilayer, and for the fatty acid tails to interact with the cholesterol of the nonpolar cholesterol tetracyclic ring here. So we have nonpolar barrier to polar solvents on either side. This is where we go with that. And now we can move on to amino acids. So here is the structure of an individual amino acid. I showed this to you earlier before. We have an amino acid group here. We have our alpha carbon in the center. We have our variable side chain R here. And we have our carboxylic acid group here. Protein structure begins with peptide bonds. Peptide bonds are always formed between the amino group and the carboxylic group. They will form this way and only this way, because the amino group will gain a hydrogen and therefore gain a positive charge, and the carboxylic group will lose a hydrogen and therefore lose its positive charge, gaining a negative charge. So they will line up along their positive and negative poles. Positive to negative, positive, negative, positive to 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 negative. They will line up in this way. Then we will have a condensation reaction occur, where we have, I'm going to shorthand this, CO minus H, and so we have here our first amino acid and our second amino acid. I'm going to combine these two. So we will have the oxygen and two of the hydrogens here combine. We'll have H2O leave. And we will have the resulting carbon and H3CO, right, NHCRHCOOH. So this is our peptide bond right there. Water left. It's a condensation reaction. We have our peptide bond forming. Let's make that pretty. That brings us to the levels of structure of the polypeptide chain. When we have these peptide bonds forming over and over and over again, they will form in a chain. The sequence of amino acids in a single polypeptide chain is the protein's primary structure. The sequence of amino acids in a single polypeptide chain. The sequence of amino acids in a single polypeptide chain. That is our protein primary structure. Which amino acid is this? Which one 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 is this? This, etc. So if this is phenylalanine, this is leucine, this is serine, this is cysteine, etc. You change the protein's primary sequence if you switch leucine and serine, for example. Or if you switch leucine out for valine, or some other amino acid. The relevant bonds of the next level of protein structure is hydrogen bonding. You have a covalent bond between oxygen and hydrogen, but you have a hydrogen bond within the molecule of water. Then you have a hydrogen bond between the hydrogen of one molecule and the oxygen of another molecule. The secondary structure takes on two forms, where you have the beta pleated sheet and the alpha helix. You have hydrogen bonding between the amino group and the carboxylic group of the members of the chain, of the members of a single polypeptide chain. Hydrogen bonding to other members of the polypeptide chain. In your alpha helix, it'll be every fourth amino acid or so. In your beta pleated sheets, it'll be every, if I'm remembering correctly, 11th, 6th or 11th, if I'm remembering correctly, of course. But this is the internet, so argue about it in the comments below. All right. Or somebody will correct me in the comments below, I'm sure of it. All right. So, the examples of the amino acid subunits, these are your amino acid subunits. There's your R group, there's your carboxylic group, there's your amino group. Look, it's a peptide bond right there. Ta-da! So, your next level of protein structure, the important bonds, are going to be your ionic bonds, your disulfide bonds, in addition to hydrogen bonds and dipole interactions. And that's going to be your tertiary structure. Your ionic bonds are going to be also called salt bridges. Your disulfide bonds will be bonds between the sulfurs. You will have an actual covalent bond, making the disulfide bond the strongest bond of the tertiary structure. Now, you can argue all day that the ionic bond is the strongest bond, but when you are in an aqueous solution, the ionic bond is a very, very weak bond, because an aqueous solution takes away all of an ionic bond's power. So, your disulfide bond, which is a covalent bond, will be your strongest bond in the tertiary structure. You also have hydrophobic interactions, where your non-polar R groups are going to dissolve and interact with each other in order to avoid the hydrophilic polar water solution surrounding and the other hydrophilic R groups elsewhere. So you'll also have hydrogen bonds of R groups elsewhere. You'll have ionic bonds elsewhere. All of those will be polar interactions. Now, your hydrophilic and your hydrophobic interactions are going to be your primary source of the tertiary structure. This is going to be where you get your main shape from. Your hydrophilic R groups and your hydrophobic R groups, your pink and your yellows. In an aqueous solution, if you put a protein in an aqueous solution, the hydrophilic R groups will face outside to the aqueous solution, and they will dissolve in water, like dissolves light. The hydrophobic R groups will internalize and go toward each other inside. So, they will nestle together, and you will have this globular shape. Now, if I were to take this same unfolded polypeptide, and I were to put it into oil, for example, or gasoline, which is a non-polar solvent, I would have the opposite effect. My pink hydrophilic groups would face inward, and my hydrophobic R groups, the yellow ones, would face outward. The folding would be totally different. If I were to dry out this protein, I would take this hydrophobic R group and I would unfold the protein entirely, because air is a non-polar medium. So my hydrophobic groups would start facing outwards. protein structure. The quaternary structure may or may not exist. The quaternary structure may or may not exist in a protein. Now this is not saying that it's theoretical. This is saying that it depends on the protein. Excuse me. So a quaternary structure is the step above tertiary structure. Your tertiary structure is going to be your three-dimensional, the 3D conformation of a single polypeptide chain. The 3D conformation of a single polypeptide chain. A quaternary structure is going to be the combination of multiple 3D conformations of single polypeptide chains into one overall 3D conformation of multiple polypeptide chains. So a quaternary structure is the 3D conformation of multiple polypeptide chains. So your tertiary structure is the 3D conformation of a single polypeptide chain. A quaternary structure is the 3D conformation of multiple polypeptide chains. Now, that brings us, and by the way, hemoglobin is our stereotypical quaternary structure. We used hemoglobin as an example. But there are other examples. For example, collagen. Collagen is a quaternary structure, but it has 3 polypeptides wound together into a fibrous polypeptide chain, which brings us to our 2 types of proteins, fibrous proteins and globular proteins. Fibrous proteins, as one might suspect, have a fibrous structure, a fiber structure, whereas globular proteins have a globe-shaped structure, a more rounded structure. So, fibrous proteins are insoluble in waters, in acids, in bases. They have strong intermolecular forces that hold them together. They are a thread or a helical shape. They are often found in sheets as well. Globular proteins, they are soluble in water and acids and bases. They have weak intermolecular forces holding them together, and they are often folded, ball-like, or 3-dimensional pattern. This is not to say that fibrous proteins do not exist in 3 dimensions, just that a sheet is not really considered 3-dimensional by ease of association. So, this is hemoglobin on a bad hair day. Hemoglobin, Hemoglobin, is the American spelling, but hemoglobin, also spelled hemoglobin, is the British spelling. I'm going to take an aside here. There's already a video on the channel about this, but about spelling. Be consistent in your spelling. Always spell different word. Always spell the word the same way every time you spell it that way. So if you're going to spell it hemoglobin every single time. If you're going to spell it hemoglobin every single time. If you're going to spell it color, C-O-L-O-U-R, you had better spell everything favor, F-A-V-O-U-R. You had better be consistent. Behavior, all the O-U-Rs. You had better be British. And last I checked, this is America. So you're not British. Alright? So try and stick to the American spellings. So, globular proteins, hemoglobin. Hemoglobin's job, and we'll talk about this much more in Unit 4, carries oxygen in the blood, as we discussed. It also has other roles. For example, carbaminohemoglobin is a compound of hemoglobin and carbon dioxide. 10% of carbon dioxide will be carried in the blood in this way, bound to the terminal amino end. To the terminal amino end of hemoglobin. Carboxyhemoglobin is a compound of hemoglobin and carbon monoxide. Carbon monoxide actually displaces oxygen from the iron of the heme group. So all this is accomplished thanks to the iron-containing heme, but that's not a protein. So because it's not a protein, but it's attached to a protein, it's not composed of amino acids, but it's part of a protein, we're going to call it a prosthetic group. So prosthetic groups may be organic, such as a vitamin, a sugar, a lipid, or inorganic, such as a metal ion. But it is not composed of amino acids. it is bound tightly to proteins. It may even be attached through covalent bonding, as opposed to coenzymes, which are very loosely bound to proteins. In A level, we would be discussing coenzymes, but this is an AS level review, so we're going to stick to the AS level right now. So this is heme right now. We have a mostly carbon and hydrogen structure with nitrogen binding to an iron compound. This allows oxygen to oxidize that iron, turning it to rust, iron oxide, therefore it will be red. So blood is always red, no matter what you see when you look at your skin and the veins on it. They're not blue, they're red. Yeah, I'm sure there's a SciShow episode out there. Once again, they don't pay me, I'm just a fan. All right, now more about fibrous proteins, specifically collagen. Collagen is a group of naturally occurring fibrous proteins in animals, most abundant in mammals. It functions as a glue that holds the body together by connecting and supporting such bodily tissues as the muscles, tendons, ligaments, and cartilages, skin, bone, and teeth. There are multiple types of cartilage, type 1, type 2, type 3, type 4. Type 1 cartilage, type 4 cartilage is bone. Type 1 cartilage is bone. Once again, comment section will probably be yelling at me. All right. Things to note. There is very little in the way of tertiary structure, 3D conformation of a single polypeptide chain here. Because effectively what you're going to get is just a strand. And you'll have it'll have a quaternary structure where you'll have 3 individual polypeptide chains wrapped together, twisted together into a cord. So collagen fibers are different from collagen molecules in a fashion similar to how cellulose is to microfibrils. So a collagen tripeptide is a collagen molecule. The individual collagen molecule is the quaternary structure of the protein. But when you combine a collagen tripeptide, collagen molecule, into multiple of its type, you will get a collagen microfibril. You combine multiple collagen microfibrils, you'll get a collagen fibrin. Multiple collagen fibrils, you'll get a collagen fiber. Now, collagen's job is the protein-based connective tissue that forms a support system for your blood vessels, skin, bones, ligaments, and muscles. It also has a partner in crime called elastin. Elastin has the job of snapping back into position, like the elastic waistband on underwear. Its job is not to stretch. Its job is to return to its original position. An elastic waistband that stretches but then does not snap back is not doing its job correctly.
Battery's out.
Battery's out?
Yep. Well, it's blinking red, meaning I'd rather cut you off now than later.
No. What?
Because it's going to...
Use it until it dies.
Yeah, use it until it dies.
Okay, sorry.
Alright. So, elastin has a similar function to the elastic waistband in your underwear, in that its job is to return to shape. So, an elastic waistband that stretches and then does not return to shape and snap back is not really doing its job very effectively. Your underwear is going to fall down. So, that's collagen and elastin. They work together. Collagen's job is to prevent stretch and to maintain certain things. So you have collagen vascular diseases. When collagen is messed up, collagen or elastin, this is also called connective tissue disorders. It will often be a group of diseases that affect your connective tissue. For example, lupus. Lupus is an autoimmune connective tissue disease that breaks down collagen. We also have rheumatoid arthritis, which can affect the collagen in your joints. We also have scleroderma, which this lady is not angry, she just can't smile because her face is too tight. Temporal arteritis, which is a problem with the collagen of the arteries, especially the temporal artery, which is right here. If that bursts, you can bleed to death. Osteogenesis imperfecta, which is a failure in the collagen that eventually forms bone. And these children, and this is a congenital inborn defect, will be born with very brittle bones, and their bones will form badly. So you will have very strange bone defects in these children, and they will have very easily broken bones. Ehlers-Danlos is a circus sideshow act from the the late 1800s, early 1900s, or even earlier than that, even the 1700s, as far back too. And this is, the elastin works, but the collagen does not. The collagen allows you to stretch beyond a certain point, and it will snap back into position. You will not be all loose and jowly. Now, this, by the way, these are all diseases that have like big problems, and they're not good for you. Like, they're not just cool. They're just not just neat. So Ehlers-Danlos has problems associated with it. Don't like be like, oh, I want to be able to do that. No. Now, let's talk briefly about nucleic acid structure. Like I said before, you have a phosphate group, you have a pentose sugar, and you have a nitrogenous base. As you can tell, this is a lot of nitrogen right here. So that's why it's a nitrogenous base. It is a base because it has an amine group and it can accept protons, and it has a lot of nitrogen. This is what you make with it. You make DNA and you make RNA. So DNA is composed of cytosine, guanine, adenine, and thymine nucleotides. Those are the nitrogenous bases. And RNA, the nitrogenous bases, are cytosine, guanine, adenine, and uracil. Uracil replaces thymine. The only difference between thymine and uracil is one methyl group. That's it. So DNA and RNA have a lot of different structural things associated with them. We will discuss those in much more detail later. So, let us briefly describe redox reactions. So, redox reactions. This is the third type of reaction that you will need to vaguely understand for the Cambridge exam. A redox reaction. Leo-Gur. If you use oil rig, good for you. I use Leo-Gur. The loss of electrons is oxidation, and the gaining of electrons is reduction. So, when you have that, if I am losing electrons, I am undergoing oxidation. If I am losing electrons, I am more positively charged. If I am losing electrons, I am less negatively charged. If I am losing electrons, I am being oxidized. If I am losing electrons, I am a reducing agent. I am the agent of reduction. I have caused reduction. By being oxidized myself, I have reduced something else. If I am gaining electrons, I am undergoing reduction. If I am gaining electrons, I am more negatively charged. If I am gaining electrons, I am less positively charged. If I am gaining electrons, I am being reduced. If I am gaining electrons, I am an oxidizing agent. So an oxidizing agent causes oxidation in others and is itself reduced. A reducing sugar is a carbohydrate that, in an aqueous medium, generates one or more compounds containing an aldehyde group that acts as a reducing agent. Remember, a reducing agent is oxidized and causes reduction. So, remembering back from way before, which are my reducing sugars? The ones that generate an aldehyde group. That would be glucose, galactose, but not fructose. Fructose is a ketose sugar. It is a non-reducing sugar. And that brings us to, before we discuss what we use redox reactions for, the testing for carbohydrates. A solution of elemental iodine and potassium iodide, also known as Lugol's reagent, but I want you guys to call it iodine test. It changes a solution that contains starch from the color of the starch solution, which is usually clear or colorless, to dark purple black, to a dark black or a dark purple or a dark blue-black color, blue-black, in the presence of complex sugars. Remembering, of course, that complex sugars are polysaccharides, the presence of polysaccharides. The more starch you have, the darker purple you get. The color change is due to iodine interacting with the coil structure of the polysaccharide. The more coiling you have, the darker you will get. Iodine will not detect simple sugars, such as glucose or fructose. But so how will we test for glucose or fructose? We will test for glucose or fructose using, using a solution that is a weak oxidizing agent. So a weak oxidizing agent in a basic aqueous solution is capable of oxidizing aldehydes but not strong enough to oxidize alcohols. That weak oxidizing agent is going to be Benedict's solution. Benedict's solution contains sodium citrate, sodium carbonate, and copper 2 sulfate. The copper 2 sulfate is the important part. It's what makes it blue. This is our weak oxidizing agent. When simple carbohydrates are heated in this solution, it changes from orange-red to brick-red. It changes from blue, this color blue, to an orange-red or a brick-red color. This reaction is caused by copper 2 ions being reduced to copper 1 ions by reducing sugars. Sometimes a brick-red solid copper oxide precipitates out of the solution. Complex carbohydrates such as starches do not react unless they are broken down through heating or digestion first. So, Benedict's solution will not detect non-reducing sugars. They will not detect starches. They will not detect polysaccharides. You must break down polysaccharides and non-reducing sugars first. For the purposes of the Cambridge lab, a non-reducing sugar is almost exclusively referring to sucrose, which is glucose plus fructose. You must hydrolyze sucrose. You must break it down into glucose, which is a reducing sugar, and fructose, which is not a reducing sugar, so that the reducing sugar, glucose, will react with Benedict's solution, and you will get this copper 1 ion reducing out, being reduced and precipitating out of solution. There is a color change pattern here. So from blue unchanged, you'll have no reducing sugars. In your early stages, you'll have trace amounts of reducing sugars when it turns just sort of green. Yellow to orange, yellow will be low, orange will be moderate, red or brick red will be large volume, large quantities of reducing sugars. All right? Anything greater than 4% should be a large quantity of reducing sugar. Less than that, and you will start to see different colors. Sucrose. Sucrose is a non-reducing sugar. It does not react with Benedict's reagent. It will not react with iodine because it is not a complex sugar. It is a disaccharide. When sucrose is decomposed to its components of glucose and fructose, the Benedict's test will be positive, but the starch test will still be negative. So, when you decompose its components of glucose and fructose, the Benedict's test will be positive because it is now glucose and fructose, and Benedict's is positive with glucose, but not fructose, but there's glucose there, so it will be positive with Benedict's. Not with the starch test, because it's still not a polysaccharide. Even though you broke it down into its component parts, you keep cutting and cutting and cutting, but your hair is still not long enough. In order to hydrolyze sucrose, we have to use hot acid, but we must neutralize that acid before adding the Benedict's reagent. Why can't we just add it in? Because weak oxidizing agents, like Benedict's reagent, require basic aqueous solutions. So if we are just adding the basic aqueous solution of Benedict's reagent to hot acid, we will neutralize that, or we will have an acidic solution. It will not react appropriately. We will not have a reduction. Our redox reaction will not occur. So, testing for lipids. These tests are not typically done by Cambridge, where you basically take some oil and you test it with brown paper and see whether or not you get a spot. You have probably held some fries in a paper bag. Yeah, that's your paper bag test. Sudan red is a fat-soluble dye that stains lipids red. It is a red dye. When you add it to lipids, it diffuses through that dye. When you add it to non-polar stuff like water, it just stays as a powder and it will not disperse. It's interesting, you could actually butter one side of bread and the other side leave blank. If you sprinkle the dye on one side and you sprinkle it on the other side, you can brush it off the one side, but the red dye will actually diffuse throughout the butter on the other side. So you'll have orange butter. It tastes fine, there's nothing wrong with it. It's just a dye. Now, this is the test that Cambridge prefers. This is the ethanol emulsion test. Effectively what you do here, you take an amphipathic substance like ethanol, which is OH, carbon, carbon, there's ethanol. So we take ethanol. We take concentrated ethanol. And we use this, which has a hydroxyl group, a polar group here, and a non-polar carbon chain, hydrocarbon chain here. We use this to dissolve the hydrocarbon chains, that is the lipids. So, lipids will dissolve in alcohol, pure alcohol. But as soon as you start adding water, and you dilute the alcohol, so that it becomes more water than alcohol. The water is polar, and the lipids are non-polar. The non-polar lipids will cause the alcohol to, the non-polar water, the non-polar lipids will come out of solution with the now diluted ethanol, and you will have an emulsion of water and lipid. So you'll get this cloudy, milky, white mixture. The protein test. We take Burette's solution, which contains copper 2 sulfate again, and potassium hydroxide, KOH. Copper will react with the peptide bond in proteins that causes the color change. It will turn purple when there are long polypeptide chains present, and pink in the presence of short polypeptide chains. So, it's not going to react in the presence of amino acids because amino acids do not have peptide bonds. You require a peptide bond for the interaction between copper 2 sulfate and the peptide bond. If it's just amino acids, no bonds. No bonds, no color change. So the more bonds you have, the more deep of a color change you will get. So, this is our color synonyms chart. These are the charts that are acceptable to use in Cambridge. Burette's test will start blue or pale blue, and it will turn purple or lilac. A positive reaction that is weak will be pink. Iodine's test will be yellow, yellow-brown, or orange, and it will turn to blue-black or dark purple. It will also be blue, bruised, brown, light purple, blah. Ethanol emulsion test will start off clear, and will turn milky, white, or cloudy. And the Benedict's test will be blue. It will change to an orange precipitate, brick red, or precipitate out copper. Other colors will be these. All right? So, we are not going to interpret those colors, and that will conclude today. So we're going to start today's lecture with base pairs, with the nucleotides and nucleic acids, excuse me. We've already covered in the previous lecture carbohydrates, proteins, lipids, and we covered very, very briefly nucleic acids. We also covered the testing for carbohydrates, proteins, lipids, etc., including the Benedict's reagent test and the reducing sugars test, as well as the starch test for the iodine's test for starch and the Burette test for protein. Now we're going to take a closer look at the nucleic acids, DNA and RNA specifically. This slide you might recognize, where we have DNA, which consists of a double helix of base pairs of nitrogenous bases, bonded cytosine to guanine and adenine to thymine. We also have RNA, which exists as a single-stranded polynucleotide, which just has the sequence, variable sequence consisting of cytosine, guanine, adenine, and uracil. Uracil is in lieu of thymine. You use uracil instead of thymine. The overall structure of a nucleic acid consists of a phosphate group, which has an overall negative charge. Your ribose, this is a deoxyribose sugar right here, because there is a lack of an oxygen right there. So this is a deoxyribose pentose sugar. And here we have the nitrogenous base adenine. You do not need to memorize the individual structures of amino acids, of nucleic acid nitrogenous bases, but this one is adenine. You will recognize adenine and guanine by their double ring structure, and cytosine and thymine for DNA by their single ring structure, and cytosine and uracil for RNA by their single ring structure. Now, pentose sugars, if you can spot the difference, deoxyribose does not have an oxygen. Ribose has an oxygen here. On the 2' carbon, there is an oxygen,